should we just go around and introduce ourselves? I think that might be quite nice. It's quite a manageable size at the moment. We can just see if, we get swamped, we can change tactics, but so far that works quite well. I mean, maybe I can just start. My name is Stegle. I'm based in Heidelberg at EMBL, the German Cancer Research Center, and we've been interested in different single cell aspects. So what we've been lately very excited about is really performing genetic analysis using single cell assays. So essentially profiling larger numbers of individuals that are genetically diverse and then linking genetic differences to phenotypic difference at a single cell level, how genes change, essentially looking at eQTLs with single cell data, but then also asking other questions like, whether we can link genetic differences to cellular identity, whether specific individuals make some cell types or not, and IPS differentiation, for example, and these sort of questions, essentially really exploiting not, one single cell experiment, but like hundreds. For many individuals, and sort of that motivated our interest in that sort of topic. So how to integrate with bulk and other assays.
Hi, I'm Jian Choi from NCI, and I mainly work on lung cancer by doing functional analysis, following up GWAS. And I have experience in building a single cell type eQTL data set in melanocytes before, and now with my new lab, I'm moving on to the lung cancer field. So I'm planning to build a single cell eQTL data set in normal lung tissues from Asian population. So we're aiming to get 120 individuals. And the main goal for me to be here is that to find some collaborators to even increase the power if there's somebody who's out there who's interested in the same topic. And also I would like to conform our analysis pipeline that are to be compatible with what other consortia are actually working on.
Mm-hmm. Hi, I'm Ayelet Segre. I'm an assistant professor at Mass Eye near Harvard Medical School and also affiliated with the Broad Institute. I started my lab about 2.5 years ago. But before that, I was part of the kind of the LDAC team at GTEx for about 5 years with Kristen Ardley and Gadi Getz. I'm sure all of you know, so I was part of the computational team in charge of the genotype QC and contributing to the eQTL analysis, and also developing methods that integrate eQTLs with GWAS. And I'm currently part of the, I have a NIH-HCA, a seed network grant to build the human cell atlas for the eye, about 10 different regions in the eye. And we're going to be doing whole genome sequence and interested also in identifying, initially, since we're only starting with 15 donors, allele-specific expression in the single cell level and eventually eQTLs at single cell level, and thinking about different models for the different dropouts and issues in single cell data, and also thinking of integration with GWAS data.
Mm-hmm.
My name is Mark Charette. I'm at NHLBI, and I'm associated with the HubMap project. And I'm very interested in expression patterns of endothelial cells. As you know, endothelial cells are one continuous layer throughout the entire body, and yet their expression pattern changes as they move from organ to organ. And I'm very interested in the question of how much does the genetic pattern have to change before we say it's a different kind of endothelial cell? Mm-hmm. Where are we going to set up those kinds of parameters, even if we, or can we? Anyway.
Mm-hmm. Good question.
Do you want me to introduce myself?
If you want, even just start being such a small group that we thought we'd just go around the table. Yeah,
I'm Deanne Taylor. I'm at the Children's Hospital Philadelphia in the University of Pennsylvania. I'm one of the PIs on an NIH project from the Common Fund called Gabriel Miller Kids First. I'm also part of the Human Cell Atlas, and I'm trying to drive the Pediatric Cell Atlas. And my interests obviously are in genetic conditions that children present themselves with at CHOP. Gabriel Miller is a genetics repository, and we're interested in how the the symptoms of genetic disorders appear in at the cell level, single cell level. So kind of all works together.
No, thanks. I think that was great to hear from everybody the different interests. So I think it's fair to say, to summarize, that this group is quite focused on natural variation, right? So nobody engineers anything. Is that true? So it's rare and common variation in assessing consequences at single cell level. So I would suggest we just focus on that subtopic, and then talk about these. Does everybody access to the Google Doc, this subtopic, genetic basis of cellular identity, that sort of sub one? I mean, it's also on the KaiStorm link where you clicked on join Zoom, the Google Doc. Did everybody access that? Yeah. So Jiayong, if you're taking some notes, I'll also try to look over and keep track. I think and we keep sort of our discussion. I think as and those who've been also participating in these discussions yesterday, there's the first part where we're supposed to come up with questions, right? And then get to the bespoke blue box in the second part. So I was just wondering, I put some thoughts up here, discussion prompts, but we can redefine them and will. And I also really like these ideas about assessing whether the genetic regulation, whether you can link variants, regulatory variants to expression changes, whether the difference in these maps is also means to get a cell identity, which I think is also really an interesting question. I think Mark, you alluded to that very, very briefly as well, right? Which is, I think, certainly quite complementary and a lot more functional than comparing blobs on a t-SNE plot and seeing which ones of these are different. So I think these are also questions we can get to, but maybe we just ask where should we start the discussion essentially. I mean, there's these questions about how to integrate with bulk data. I think there's a question about also cell identity. I'm just writing that maybe here as well as a sort of cell identity link and in relationship to EQTL maps. I think that's maybe the way to put it. Where do you want to start? What excites most people?
Well, I can just mention that one of the things that excites us is in diseases of inborn errors of metabolism, cell identity doesn't necessarily change, but we think the cell functions radically change. So, and shows up as a disease phenotype. So I don't know, maybe just, that's what excites us anyway.
So can you just unpack that a little bit? So you're asking how the regulatory changes are.
Yes, how do regulatory change, exactly. So regulatory changes in cell types. We can't tell the difference yet in the community, I believe. I mean, although there's ideas between cell type and cell state, right? So there's ideas of like, does an activated T cell, is that the same type of cell as a T cell, or is it a different, in a different cell state, or is it a different cell type? So we've noticed that there's different cell states in kids with some, and this is stuff that people are working on now, but that the cells shift, in gene expression when there are some specific types of metabolic lesions. So one of the things we're interested in is, are the cells shifting and not really changing, well, they're changing state, but are they changing state and somehow affecting the cell type? But then we're seeing some cell types not shifting as much. So it's sorry to be so cloudy about it, but we're just trying to identify, is there a difference in cell state that can be quantified by a genetic lesion? If that makes any sense. So in other words, does the same genetic lesion always yield the same shift in cell state in the specific cells?
Yeah, so it's case control associated changes in cell states, right? And and you ask to what extent and the relationship to genetics, and relationship to genetic factors. I think that, I guess the question is, which ones of those are genetic and which ones are not, right? That's also probably,
right. Yes, that's what I'm trying to get at. Sorry, as Sarah was saying earlier, I'm being a bit of a muppet this morning because I haven't had caffeine.
No, I think this is an exciting question. Yeah, Mark.
You know, it seems like our interests are kind of disparate. You know, it's hard to find a common theme among the various interests of this small group. I mean, my interest in endothelial expression patterns comes from my also interest in being able to find a common coordinate framework. I mean, the shift in endothelial expression patterns as it moves from organ to organ could be diagnostic of position. You know, if you think about it, you know, a map of the endothelial expression patterns could point to where you are localized in the body, you know, and as you move from position to position, there's data in the brain system, in the brain, which shows that the endothelial expression pattern, as it moves from region to region, changes in a very smooth kind of continuum. It's not necessarily linear, but it moves in a continuum. And that suggests the possibility that if you know 2 points that are separated by some distance, you could use computer algorithms to determine all the intermediate points, you know, and as you gather more data, the system would become more refined. So anyway, that's sort of where my interest in it lies. But again, I'm not sure exactly how this group is going to find common ground to be able to suggest something of a uniform stature when we report in.
Yeah, I agree. That's very difficult. But you also mentioned this idea of, you know, linking genetics to cell identity, right? Is that linked to that? I mean,
absolutely. I mean, it's how you know, an endothelial cell in the brain is very different from an endothelial cell in the kidney, and yet how much of a change has to occur in order for us to say that this is a distinct cell type? This is going to be a real problem in endothelial cells. You know, at what point do you say this is a different endothelial cell from that? And I don't have a good answer at this point in time. I'm not even sure I have a good approach to the problem.
Mm-hmm. Mm-hmm.
Sorry, I had to step out, help my daughter get set up for an online class. But one other topic I thought maybe to raise that we're interested in is thinking about the cell type specificity of genetic regulation of gene expression and how this compares to tissue specificity, and gets a bit, Mark, to your interest of, you know, an EQTL that's shared across tissues, maybe due to a cell type that's shared or a shared biological process, but also to think about, you know, if it's worth also comparing what we're going to be seeing in the single cell level for genetic regulation to different, the methods that people are currently using to compute cell type interacting EQTLs from bulk tissue, from deconvolution analysis of estimating proportions of cell types from bulk tissue, and then looking for EQTLs that interact with the proportions of cell types. So thinking of you know, comparing EQTLs that we identify at the single cell level to ones we identify from bulk tissue.
I think that's an interesting topic, and then it also loops back to identifying the cell type of origin in some complex common diseases. For example, in lung, there are like over 50 types of cells, and there are many histological subtypes of lung cancer, and they're originating from different cells, and then how different cell type EQTLs could specifically contribute to disease etiology in different histotypes would be an interest and connect to that point. Yeah,
no, I think that's an interesting question. I think we're getting there. There are some themes that we have shared interest. And I think the cell type specificity of genetic regulation, you know, and I also like the idea, you know, this could be both cell states, but also cell types. The cell type is maybe classical, conventional, but also cell states. And effectively every cell is a combination of those, and they probably jointly determine the regulatory profile. And there's a combinations of discrete versus continuous. I think that's a great way, and then the integration with bulk and cell type fractions and other elements. Yeah. Are there other questions? Maybe just gathering, I guess, ideas right now, right? And then we can go into the more, specifics later, I guess. Actually, how long do we have? Actually, just tracking. But I'll find that out.
I think 45 minutes for this session.
Yeah.
And a break, and then 75 on the other side.
So it's at 5, right? At 5, we're sort of done. No, yeah, 5 CET, so 15 minutes. Yeah. And then we'll break. I guess we just want to, if we have other questions that we want to go into.
How do we, have anybody else sat in on any other meetings about truly, well, not truly, but accurately expressing cell phenotypes so that we can generate a kind of identity of a cell? Because we kind of all have our own concepts of what a cellular identity is, but do we have a phenotypic definition? I know that this has been people are working on this in CellML and other ontologies, but and are we identifying cells simply by their expression pattern, the cell type? Or are we going to be trying to look at a deeper idea of what a cell type is? So for example, we have cell types that you can identify, or cell states you can identify by clusters and expression data, but if you added a orthogonal axis like proteomics, you might find out that you're really looking at 2 different cell types or more that are differentiated by additional data as it comes in.
I can weigh in on this because I've been in several sessions that have asked that same exact question now. I've been a moderator kind of skipping around. Mm-hmm. And the short answer is nobody has aligned on that. Everybody aligns on the fact that the transcriptomics profile is the easiest one. And then when you have it and as optimized to the different cell system, additional data is nice to have. So coming from the brain side, we rely very heavily on other phenotypic profiles that are not just transcriptomics, but morphological as well as electrophysiological. And everybody
Definitely
seems to be seeing all of those things as second order. But it's coming up now in every single question. Many, many different groups are asking that same question.
Yeah, because when we align strictly on metabolic genes, we end up with very few clusters, and it's either because you have so few genes in your collection of expression data, that they happen to make the clusters that they do. So expression data is a very, very limited canvas, as you said. Okay. So,
but I think there is an interesting dimension, just to maybe quickly jump in there. And I think that's very quite specific to this session potentially, right? That's, whether putatively different cell types, if you call them like that, we have from the transcriptome, whether the changes in regulation is one of these other dimensions that you could do, right? I mean, you can sort of populations to whatever, proteomics, metabolomics, whatever on them. But there's obviously the alternative as well, that you look at many, many individuals, ask, are there changes in the regulatory landscape? Yeah. And I think that's maybe one contribution that this could give, which is actually closer to function, definitely closer to function than just looking at a transcriptome pattern. Yeah. And I think that's maybe an interesting angle, right? Particularly if that's a common theme, it might be useful to sort of make that point, I guess.
Well, I think that's a really good point. Do we have any good examples of cells that are showing fundamentally the same transcription profile and yet are differentiated in some respect? I mean, via proteomics or some other parameter? I mean, is anyone aware of any group of cells which shows fundamentally the same pattern, but can be differentiated into 2 groups based on something else?
I can do that because like I mentioned, if we limit our clusters to just metabolic expression, we get a lot of cell types clustering together. Whether or not that's due to insufficient amounts of data, because we're only clustering on 2,000 genes rather than 20,000 genes, that's a hard question to answer, I guess is what I'm saying. Because I do get mixes. I'll get like 3 major or 4 major clusters from metabolic state if you assume that the expression patterns for metabolic genes are metabolic state. And when you look at the data, you're seeing mixes of different endothelial, epithelial cells in different clusters, but from different organs. So if you separate them on higher dimensions, right, with 20,000 genes, you start getting better clustering. So if I had potentially proteomics data ended up in there, could I cluster them even further? Possibly. So the clusters are always arbitrarily assigned by similarity into these ideas of meta cells or whatever they call them, but there's no, I don't know if those are really data restricted, I guess is what I'm trying to say. The data is restricting the clusters.
I wonder if this is an example of where machine learning could help us. You know, I mean, if machines are now able to look at retinal patterns and say whether it's a male or a female, I mean, maybe there are patterns within the transcription data or the metabolomic data that we simply aren't seeing at this point in time.
Yeah, I think there genetics can't really help, right? I mean, you know, I mean that's probably just hard to do, right? I mean, it's true that there could be beyond RNA. I mean, we could also think about a topic about beyond RNA genetics, right? That's the same question that you're posing on cell type identity. We could ask, you know, do we do things like proteomics and cell metabolomics? I mean, maybe that's also a question. Is that maybe a way to tie it to the topic?
I just think that genetics has different effects on different systems. And that's my opinion, but for example, metabolic changes, you'll see specific types of systems being changed rapidly and others not so much. So I'm wondering that, you know, and that's just an extreme form of expression change, I think, is when you start really changing the fundamental energetics of a cell. But in other examples where you would have small changes like EQTLs, where you have to start changing expression in the single cells, then one of the things we haven't discussed is allele-specific expression that would affect expression in the single cells. And I'm not sure if people, I think someone's done something like that, right? Looked at allele-specific expression in single cells. But that's another way to look at these genetic changes that'll be more subtle, that would change expression from cell to cell or cell type to cell type.
I mean, I think that's a technological question, right? I mean, you could do in principle, you can do the allele specific or population, you know, fundamentally it's the same thing, but I agree there's an appeal that you can do with smaller sample sizes. And, you know, I think it's a nice technological aspect, leveraging ASE, which I completely agree with. I mean, the downside obviously is that you probably need full-length transcripts. It's much harder with 3', 5' seq. So, you know, it's not.
Do you suppose we're missing part of it too by not looking carefully enough at the mitochondria? I mean, we have a tendency to look at the nuclear DNA and the nuclear transcripts, but a lot of disease is associated with mitochondrial defects. And we generally don't focus in on that.
Well, interestingly, I know in brain, we sort of did the inverse of that, where we have done at the Allen Institute, we've done a lot of single cell profiling as well as whole cell profiling. And we see discriminating differences when we're looking at one modality versus another. Some of those limitations are just practical in terms of what kind of tissue is easier for us to access. And actually, when we're layering on multimodal analyses of single cells through things like electrophysiology and morphology, the reason why we do nuclear profiling is because we can only access those other modalities if we're looking at the nucleus and not sacrificing the whole cell. But we do see profile changes that will shift when you get, you know, just I think as Dee, I don't know your first name, but Dee Taylor, as you were saying, when you get beyond some of the low-hanging fruit of genetic markers or transcriptomic markers, you start seeing the differentiators below that. So if you're looking at the most highly expressed or the ones that allow that cluster to form, you're seeing homogeneity. When you start looking at that next level, you see heterogeneity. And we definitely see that when we're looking between whole cell RNA transcript versus just nuclear RNA transcript. And that is where you start seeing discriminators in certain either closed cell types or cell types that you sort of know, or you have an intuition, or based on their function or appearance, you know that there's something else that's different, but just looking at your transcriptional profile, they appear the same. Does that make sense in the context of what you were talking about?
What about you know, sort of other data modalities to define cell types or cell states that you can then assess genetically, right? With multi-omics technologies, we could look also accessibility, and then, you know, then define cell types, cell states, and then look at what genetics does in transcriptome, or DNA methylation, or, you know, whatever is available, yeah, essentially. And do we feel? I mean, essentially, it's this beyond RNA question, right? What other assays should one consider? Is that a
Of course, it's DNA somatic. You know, it'd be nice to look at DNA. I'm not sure how well technologies work right now for RNA and DNA simultaneously. But it would be nice to look for somatic changes as well, even in cells that are not cancerous. We see these, for example, in cells with anemia. There are somatic changes. So it'd be nice to have DNA, and obviously proteomics someday.
I mean, somatic change is something we didn't really discuss. You know, this is all germline so far, almost, right? I mean, that's maybe a different question, yeah? I mean, identification of somatic changes in single cells. I mean, that also, you know, there's a whole question about how to do this experimentally, targeted DNA, and, you know, that's a...
Yeah, because I think Chris Walsh showed that, I think he proposed that some epilepsies were due to local germline changes, right, to the brain. That was a while ago, and I'm not sure what happened with that study, but with anemias, there's also local somatic changes that are not cancerous, but they just have different changes in expression that they went and found out that they're probably somatic DNA changes that you end up with these clonal expansions. So yeah, that would be great. I'm not trying to leave out cancer. I'm just saying that on top of cancer, there are other diseases that are related to kind of like a local germline change.
What about adding also DNA methylation status association with expression or other modalities?
Yeah.
So let's say, I mean, there are a lot of methylation QPLs, and those tend to be more stable. I think it's just a matter of being able to assay these, measure these, and get these assays in your tissue or cell types of interest. But there are data out there, I'm sure, for DNA methylation, and the somatic mutations. We're, for example, planning to do a whole genome sequence on the in the eye atlas, and we can choose. We're thinking of choosing one common tissue target region to do the sequencing. So the idea is to use it as germline mutations, germline variants, but we could also extract somatic mutations from that.
Yeah, somatic changes in single cells. I guess what you're sort of alluding to is also, from different assays. Yeah, so this could be obviously specific DNA-seq, but also RNA methylation, etc. Accessibility attack.
Right.
I think that is a question, how to get these variants out, and then obviously you can ask what are the regulatory changes. Yeah. I think it might make sense to split this up by germline. I mean, there's sort of 3 dimensions. One is germline versus somatic. The other one was also rare versus common. There was a bit of a thread before as well on looking at rare patients potentially, and rare disease genetics, which I think is quite a bit different game than doing sort of GTEC style population variation. do we want to tap this rare disease question, or do we carve that out? I mean, that's probably also a question how many things we can get into.
I just want to mention that the NIH has got a new call out for Mendelian genomic centers for undiagnosed diseases like rare diseases, and they're going to be funding a series of centers to do exactly that, to look into, to try to solve these rare diseases using genomics, single cell, things like that. I can paste in the RFA. The due date's July, but there already existing Mendelian genetic centers that are going to be applying for that, and that's a really large consortium of possible subjects. Yeah.
Yeah.
So maybe another topic is just the genetic regulation of what phenotypes. Obviously, gene expression and proteomics is starting to emerge, but to think of other phenotypes, you know, chromatin structure or histone modifications. Other, I don't think of higher level, just a thought, not something we've looked at yet, but if there's any genetic regulation to the cells, the distribution of cell states that you see in individuals, so switching between cell states, if there's any genetic basis to that. And then the cell type specificity aspect of all of this.
Yep, well, that makes sense.
No.
I mean, and another dimension, I'm just wondering whether we should discuss it. I mean, this NIH activity was, I think, an exciting prompt. You know, I mean, what are the data resources to do this, right? I mean, you know, we talk about modeling and integration, but one of the integrations is actually to pool data. There's now efforts like, you know, Louis Franke kicked this off in the blood context to, you know, pool single cell EQTL data in PBMCs and thereby get more sample sizes. So I'm wondering, should we have a section on that? What are data resources that what could tap into, or what are the challenges in doing so? I mean, as soon as you want to share these types of data with genetic information attached to it, it's obviously a data protection issue as well. And, you know, is that something we want to get into? And
having population diversity and then just combining resources might be, it could link to the power in detecting genetic influence in all those cell type, state, and disease. So maybe some aspect of that.
You know, Oliver, one of the interesting things about Gabriel and Miller Kids First is a genetics repository is that it has such a broad range of different birth defects in it for studies. They're cohorts of trios. And there was a guy up in, and they're kids with either cancer or congenital heart defects. And there's a guy up in Toronto, Mark Taylor, who was able to show recently that possibly a lot of pediatric cancers are germline, believed to be germline based. And he showed that there are cell types associated with medulloblastoma that might be arising because of incomplete migration during development or some kind of incomplete differentiation during development. That was like last year he published that. And there's a definite genetic link between pediatric cancer and birth defects. There's a high risk of kids with birth defects, a higher risk of kids with birth defects getting pediatric cancer. So there are single cell types that may incompletely differentiate or migrate due to genetic factors. So these cell types don't completely reach their intended state or type. So there's that development-based genetics single cell link as well.
yeah.
I wanted to mention it last time, but I also did not want to dominate the conversation because I'm so excited about this topic that I will not shut up sometimes. So I'm trying to shut up.
I think that's a fundamental one. I mean, leveraging single cell readouts to assay genetic effects on developmental dynamics, right? And cell and lineage formation. I guess that's what you're sort of getting after. Yeah, I mean, we have some data sets where we use IPS-based resources and, you know, differentiating large panels of IPS cells where you sort of have a model for these early developmental processes, and we see that in a dish, right? But obviously there's all the in vivo side, and yeah, that's exciting questions. Are there any more thoughts that we'd get into with that before we go into the second part? I guess we should sort of really think about more with the second part, right? Which is sort of recommendations for collaboration and address the problem and take it forward. I guess I would just quickly move there to start addressing some of that. You know? I mean, my thinking was, I mean, based on the previous session, but, you know, please do jump in any order that you think makes sense. We could go after. Would be that we can order this by germline versus somatic. That seemed a very interesting angle to, you know, and then there were different dimensions we felt about and challenges. One of them is, you know, giving novel technologies, where ASE was something that came up. Other data modalities, the integration with other data modalities as bulk data and so forth. So it's a bit of a question how we want to come to these recommendations or collaborations that might address problems. Yeah.
Is there a way we can use models of, you know, generate stem cells from like LCLs or epithelial cells from kids with genetic defects and create organoids or something? That's something that we were thinking about doing in terms of specific tissues like retinal cells or something like that for kids with specific mitochondrial diseases. Is there a way to create models? I mean, you could use CRISPR as well, right? But it's not going to have the same genetic background as the kids with these. So, I mean, there are people with repositories of these epithelial cells or LCLs, and there's ways, right, obviously, to turn them into stem cells. So that might be, it's not so technological as much as it's experimental, but that might be another interesting way to get people to start working on the genetic link to cell type at the single cell level. I know that there's some protocols, very good protocols for certain organoids like heart, right? And some others that people have characterized. I'm wondering if that would be a good experiment to set up among a bunch of different genetic backgrounds. And then look at single cell behavior to those links to genetics. I don't know. Because we have resources here for mitochondrial diseases, especially with kids with specific severe, you know, they have severe phenotypes. And there's definitely different tissues involved that are specific for this particular phenotype, and they're not solved diseases. We just don't know exactly what to call the disease. Some of them are pretty unique. So there might be ways to study these diseases through the single cells. And so I'm not sure if anyone, I mean, that might be a way to set up a small consortium of people to study this at a single cell level, you know, specifically for, you know, diseases that manifest in pediatrics. Of course, they're lifetime diseases. You just detect them first when they're kids.
Yeah.
Yeah.
Yeah. But do you think primarily about developmentally manifesting disorders? So
they're developmentally manifesting, but some of them show up. I mean, the kid looks otherwise. I mean, sometimes you can get a thing where I mean, even there's some examples of metabolic diseases where the kid is mostly healthy when they're born, and then they go downhill right after that. So they're not, if you look at them, they don't look developmentally different. It's just that they're going to be developmentally different really quickly because their bodies aren't making the same, they're not sustaining their system because they have a metabolic disorder that the mother's placenta, the mother's system was able to supply for them, but on their own, they're not able to. So there's a bunch of different types So there's ones where there's the metabolic functional diseases, or diseases of like Tay-Sachs diseases. So they go beyond structural birth defects. I mean, autism is one where people think that the child is, although this is up for debate, but people say that, their kid is mostly normal until suddenly they get vaccinated, which happens to be about the same time that they do get vaccines, that they start manifesting symptoms of autism. So, I mean, that's obviously probably something that shows up early on in later in development or early on right after birth, but no one's tried making mini brains out of autism kids and looking at single cells yet, I think, unless they have, but I haven't heard of it. So there's a lot of diseases we could study to see how it's manifesting at the single cell level without having to do, get an autopsy on these people.
Yeah. So the sort of iPS-derived models for capturing that. I mean, there's another dimension, we could get to that, but is also, well, that you could also consider pools, right? I mean, we're starting, sort of looking towards particular iPS-derived model systems where you look sort of compound organized that consists of cells of different genetic background, right? And so intrinsic genetic effects you could monitor quite effectively using these sort of systems, and I think that will really take up. I mean, but would it be an interesting dimension, to just say recommend that, that for many of these experiments, we should just in general introduce population variation as a dynamic into experiments that we do. That could be a generic recommendation that one could do. And then we could give a few specific examples. In iPSCs, that's obviously an obvious question, but it could obviously be in other contexts as well. That one can revisit that dimension that we at the moment ignoring in the atlases. That might be a very specific thing. Should we?
Go ahead. If someone else wants to say something, I was just going to really quickly say that there's a lot of diversity. So I'm not sure. I mean, you need a lot of samples. I think Gian, right, said that earlier, that you needed a lot of samples when you start looking at with population diversity.
Yeah, I was just going to add the same. I think that would be great recommendation, but probably not something that would be feasible to do initially, at least. I think eventually, for all the tissues and organs, we'd love to get there. But at least I think also, as Oliver, as you said, to start thinking of what data resources that are out there, thinking of what tissues, or of course in blood, there are already efforts to collect and do single cell RNA-seq at least across multiple individuals. I think it would be great. I think based on, I think Lou Defang showed that he could get single cell EQTLs from as few as, I think, 45 donors. But that's probably really the lower bound, 45, 50. So I think it would be a great recommendation, at least to start looking at that, more at that axis, and whoever can. I mean, with the human eye atlas, cell atlas, we eventually want to get there, but we have funding for initial 16 donors. So that would be the goal, and probably we'd focus initially on one region or two regions to go the axis. And so allele specific expression, for example, can start getting a bit at, it's limited again because most of the RNA sequencing is 3' or 5', but can start in parallel also to look at genetic regulation in a smaller sample set of individuals.
Yeah, so I think that's a good point. And then related to that, I personally had a hard time identifying from each consortium how many samples of which population they have collected and that they're aiming to collect. So obviously accumulating a lot of samples and generating data takes a lot of time. So in the meantime, if people kind of know their goals, then they can work together in the process to eventually, even if you cannot harmonize the data analysis process, you can run something like meta-analysis using summary stats or something like that. So I think kind of sharing that kind of resources, like even their plans and a number of samples, would be beneficial to the community, I think.
Yeah, I'm just trying to put this into something specific and right. I mean, the first one, I guess, and just to capture it more abstractly, was that we, I mean, maybe if you also in this document, is really that we want to introduce genetic variation component into various systems, experiments, and systems where it makes sense, obviously. You know, one was IPS derived, so this could be organized, you know, considering also pooled experiments, that would be an option to do that. You know, I think, obviously, if you think about common variants, you know, tens of individuals is already enough. You know, I think like 100 IPS lines that are pooled, one could probably, in quite a few experiments, do quite a bit of that. There's also existing efforts, you know, including something like GTEx or rare disease consortia, where one could basically have a single cell component. That's sort of what we're sort of proposing, I guess. And the second, what I'm hearing is sort of methods and approaches to maximize value and mitigate the typical small sample size, right? I mean, we will have small samples in principle. And so we need methods that get the most out of this data, even though we will probably be bound to like 50, 60 samples in most cases. Is that a fair point? And there, you know, we had two approaches. One was, you know, using allele-specific expression and methods specifically suitable for single cell data sets of this type, of this nature. You know, that's something, I guess, where probably needs more developments. I mean, you know, I think quite a few groups are thinking about this question. We talked about integration with bulk as well.
Uh-huh. And there's lots of tools like Magic that can deconvolute bulk data as well.
Yes, I mean, I think that's a really good point. That I think the two questions with bulk. One is bulk EQTL maps, right? You could basically say, if you know already that variants are regulatory variants, you know, you can utilize this in a, you know, your multiple testing adjustment and other elements. One of them, revisit existing variants, but I think, you know, you can't afford to ignore the data when you look at these single cell data. It just would be because of power, e.g. to maximize power, you know, multiple testing and so forth, yeah. And the second, I guess, was, but maybe it's the second category, right? Is using small single cell RNA-seq studies to enhance bulk EQTL analysis, e.g. via deconvolution. Yeah, is that maybe another?
Yeah.
Yeah.
If you have that, and I think you can get that from the old GTEx. You might be able to deconvolute cell types, so then you could look at if there's any influence of genetics on any kind of cell types. But this is all modeling, right? So it's kind of one step away. Another thing, can I request in this is that have we ever done, in interest of population studies, have we ever gone through the Human Cell Atlas and done a survey on what kind of genetic diversity exists right now? For example, I know that Sarah Tishkoff, I don't know if you've heard of her, but she's done about 5,000 or more whole genome sequence samples from people across Africa. They have East Africa and West Africa mostly. She's actually gotten a CZI grant, I think, to look at some single cell data from the blood. She has blood samples from a lot of African tribes. So that's like, I know that exists, right? And I know that China, there's a Chinese group, a large Chinese consortium that just released a whole bunch of single cell data, right? So I'm wondering if there's a way that we can put together a list for those population and genetics people to find out where the resources are and what kind of ethnicities exist. I don't even know if that's one of the, is that one of the choices on the Human Cell Atlas browser? The portal browser? Does anyone know? Because otherwise I can go look.
I don't know, as far as I could tell, I couldn't find anything about population other than they're promoting diversity in a much smaller group than GTEx.
It would be good to actually have that. Here we go, tissue type, donor. Let's see what we have for donor. No, they only have sex, species, age, and unit. I mean, we can request this. We can request them to include, I mean, we can also try to determine, I mean, if you have their genetics, but we don't. So we really need to know their self-reported ethnicity and race. Race and ethnicity would be nice to have. So that really should be a priority for this group as well, right? Is to try to make sure that that's included, at least in the different consortia. So not even just the HCA, but in one of the other sessions, they're talking about Atlas integration. And of course, there's the metadata group as well. It'd be really nice to have that information be required as a standard. I mean, even if it's generic, like this came from Africa, and then we could say, okay, at the least, the most we have is that this is an African population. We don't know what part of Africa. At least we know that they're from the African population. It'd be nice to have something like that.
That's population metadata. I mean, one could even go a step further and say you should just genotype every sample.
Mm-hmm. Oh, that would be wonderful. Then you could really look at allele-specific expression if you had enough data, right? If the data was, if your reads came across and hit one of those.
Right? I mean, if the ethics for the HCA would require that we can do genotyping, you do that. Run a genotyping chip that's neglectable.
Well, in pediatrics data, we have genetics data that can go with the single cell data, and HCA is setting up a part of the portal that has protected data mostly because of the European requirements, right? The GPC, I can't remember the name, but you know what I'm talking about, the restrictions on privacy. And so that part of the portal supposedly exists. There are ways to get around that and to make sure that the data is protected and that people would have to request it through a data use committee, so that you can have that data there and maybe do summarizations of the data without having to use the protected data. But you could also request the genotypes yourself as part of a data use if you had single cell data and genotype data. I'm just saying, I don't think it's impossible, and it would be a really nice addition.
Well, that's extending, what do we say? That's extending the HCA data, you know, data interest mechanism and optional / required filters, that there is a mechanism, you know, I mean, you can imagine if everybody now deposits God knows how many HCA data sets, and there'll be hundreds of blood samples and God knows what.
Yeah.
People include genetic variation panels, you know, one would obviously be able to reanalyze a lot of these data later on and do these sort of things. Is that what we want to propose? I mean, that's very specific.
That would be great. I mean, there are some panels that don't expose the individual, hopefully don't expose an individual's identity, but still gives you an idea of their ethnicity. Correct? Because we worked with some of those before. There are a limited set of panels that just give you common variants associated with different geographical locations. So that would be, yeah.
Ethnicity is easy, you know. I mean, consider record linkage of open access. I mean, I don't really know. I mean, you know, in a way, if you have an ethics that allows you to share the single cell RNA-seq data openly with the rest of the world, you essentially can also share the genotypes, right? I mean, I don't see that many ethics committees have an issue with that, because, you know, you can reconstruct pretty much. I mean, far too much from that read data anyway. So
yeah,
I mean,
that goes back to, that's a really good question. That is a really good question.
And from a session yesterday, there was a question raised about getting IRB and how that connects to sharing the raw data from a single cell RNA transcriptome, because you can use coding variants, as you said, and track back, you know, the identity. So even though those samples are de-identified, sometimes you need to get consent for sharing that information later in the process. So I guess it could be a little kind of a tricky issue about the genotype and then even the transcriptome data.
They probably mentioned it. There's there are computational biologists who can strip identifying alleles, basically replacing all variants with the most common allele, which removes all ability to do allele specific expression if you wanted to do down the line. But there's some people who've proposed, if maybe not in these sessions, but having two different tiers of data. One is data that's been stripped of all identifying variants in the single cell data, and then another tier where it's identifiable that you have to apply to get the data. But that kind of ruins any genetic studies on exactly what we're trying to do. So we just to even look at genetic effects, we have to have data that is either protected and we can apply for it, or data that is open. So
yeah, I mean, I think it's definitely something, you know, to really think about, right? Augmenting HCA with a genetic variation component. And as you're saying, there are sort of two options to this. One of them is, you know, I mean, different stepwise, you know, simple uploaded data uploaded, meta information on ethnicity. I think that's sort of metadata on ethnicity. That's probably quite straightforward. And then more complex is either record linkage with genetic repositories, controlled access genetic repositories, and or the incorporation of genetic data in the HCA portal itself, right? I mean, that's something one could also consider, right? I mean, you could do record linkage with dbGaP or whatever. You have to say this is an HCA sample and the genetic information is in this other repository, and you can do that. I mean, that would be. It seems like it might be an opportunity if we miss out on this genetic data later on. We can't ask these questions that we would otherwise want to ask.
Can we add and other consortiums with HCA? Because this includes NIH, these meetings also, and so like HubMap and stuff. So I would say considering augmenting all single cell consortiums or.
Yeah, right. I mean, sorry, that's a very good point.
No, it's all right. I'm biased to HCA too, but. I'm just thinking. I just saw one of the screenshots from one of the slides from Sarah of all the different things going on, and there's a bunch of different single cell projects. So I figured, yeah, there you go.
Yeah. I mean, I think I think that makes a lot of sense. Yeah.
Yeah.
Okay, so this is already, I mean, one is more data generation, where I think new design should be efforts that consider a genetic variation component in different contexts.
Mm-hmm.
Experiments and systems, so that I think makes a lot of sense. And then we have IPS-derived existing efforts, including existing population studies. Maybe we can say that, right?
And CRISPR, maybe some CRISPR knock-ins too. I mean, for dominant variants. I mean, that's another option with these experimental systems. I don't know if you want to explicitly name it, but
the disease consortia, both patients and CRISPR.
Right.
Yeah, I mean, that's either you directly use patients or CRISPR, particular variants of interest. I think that makes sense. And then it's consider augment HCA and other single repositories with genetic variation components. I mean, that makes sense. Simple metadata, more complex record linkage, controlled access, repository, and/or incorporation. I think that makes a lot of sense. And then the third thing we have was methods and approaches to maximize value, mitigate too small sample size. So a little specific expression was something that we discussed. And there are new methods available and needed, and integration with bulk EQTL maximize power, multiple testing from when considering cell types with existing EQTL resources. I mean, bracket open GTEx. It also is blood is a very important example, right, for many of the questions that people will be interested in. And then we also mentioned to exploit smaller single-cell RNA-seq studies data to enhance bulk EQTL analysis. Yeah, for example, via decomposition. I think that's a quite nice package already, of germline recommendations. So anything else? So specifically, already, what do we want to say here as a specific recommendation? I mean, we haven't really mentioned the sort of method side for looking at cell type, cell state. We discussed this before, right? To sort of lose the genetic variation component for solving cell identity. I mean, that we could also do with somatic variants, but I think the germline component is probably going to be easier, right? To really say, can we use this to redefine what is a cell type? I think there were lots of comments from quite a few of us here in the group to mention that, and so maybe
there's that multimodal multi-omics meeting going on in parallel to this one.
And they probably also say that they want to use multi-omics to identify cell identity, right? It makes obviously sense.
Yeah.
I mean, but we could say that it's another layer we should add to this, yeah? Consider regulatory, exploit, how do we call this? Regulatory variation to address core questions in single cell biology. And the one question we wanted to we specifically said is cell identity. Yeah, I mean, are cell types defined by distinct regulatory dependencies? This is towards a more a functional definition of cell identity. I think we discussed that, and I think it makes total sense to put that forward. And the other one was sort of also, you know, this discrete versus continuous. I'm not sure how we want to capture that. You know, we discussed this.
I guess we were talking about cell context at one point,
yeah,
he was talking about the endothelial cells being in different contexts, changing kind of their expression identity, even though they're all supposedly the same cell type.
Yeah.
I'm
wondering if we should do this to 3 and say it as methods and approaches for single cell EQTL, single cell genetic analysis. Maybe we want to do it here, you know, because essentially, I mean, that's an opportunity that these data have. You know, one is using this specific expression, better incorporation, deconvolution. The second was sort of modeling the individual determinants of regulatory landscapes. Yeah, and that's cell type, cell state, etc. Context in general. Right? I mean, we could tease it apart. Yeah, modeling. I don't know. Is that a fair point? Is that a maybe okay summary? The integration. Methods.
I think it's a small point, but in thinking about kind of in the point of leveraging, actually now you've changed it. Yeah, leveraging the single cell RNA-seq data to enhance bulk EQTL analysis. But I think also just in general, evaluating how the genetic regulation will be detecting at the single cell level, how it compares to methods we're using currently with bulk RNA-seq data to identify specific cell type interacting EQTLs. To what extent we're detecting with single cell data regulation that we can't detect yet with kind of deconvolution and cell type interacting methods.
So you want to contrast that, right? I mean, there should be we should contrast that and get more insights into this.
Yes, yes.
Explicit benchmark or comparison.
Right, a signal on a seq EQTL as opposed to bulk tissue deconvolution and interaction methods.
Yeah. I think that's a very important point, right? I mean, you need to know what's the added value, right? At the moment, I think there's no clarity on that. Yeah? We know we can rediscover the same things, and you know, but I think a very
Studying that in GTEx, sorry, just one quick point. In GTEx, we're finding with the cell deconvolution analysis and cell type interacting QTL analysis that was done, some of the cell type interacting QTLs are not found at the bulk tissue level. And then the question is, you know, looking at single cell level EQTLs, if we'll still further identify EQTLs not found with the bulk methods. Probably a lot of it has to do probably with power, but it'd be good to evaluate the added value.
Yeah, does it also go in experimentalism? You know, I mean, for some questions, it might just not be the right thing, right? I mean.
Right. And do we want to add here also genetic regulation and gene expression? RNA-seq is the first obvious one, but do we want to add here also looking at genetic regulation of protein levels and other phenotypes that are histone modifications, other assays that are being done, other phenotypes that are being measured, molecular phenotypes that are being measured.
You can even use ATAC-seq for that, right?
Yeah. Shall we do this in the new experimental data generation efforts on this point 1? Does it fit there?
Yeah, sure. Or add that to existing, to add that to recommendations or consortiums or.
Yeah, consider non-RNA, beyond RNA single cell assays. I think that makes great sense, right? I mean, you know.
Yeah, for genetic effects.
So that's also an interesting question, right? I mean, you know, at some point we will be able to do spatial transcriptomics and lots of people. It will be quite a fun world. Yeah,
it would be really interesting to see if the structural and expression and proteomics differences fish. Because you can get that with histochemistry, right? You could start looking at at least on the surfaces. You could look at I mean, we're interested in epitopes at CHOP. That's why I'm thinking about that. We're interested in, I know that's often for cancer studies, but you're looking for genetic differences that predict whether or not a certain, so let me go backwards for a minute. So there's a lot of cell surface targets that people use for pediatric cancer targeting, like with CAR T-cell therapy, but we're not yet aware of what children and what tissues and what children are going to be expressing the same targets on normal tissue, which would make it a bad choice for a target, because you don't want children's hearts being targeted by a T-cell therapy. So there may be a genetic component as well as to what epitopes are expressed on the surface of cells. So this is all stuff that people are now actively either working or considering it in the pediatric level. If we're going to target a cancer cell, how much of a mortality or morbidity are we going to get with these kids based on our target choice?
Yeah.
Yeah. Can we also add something to number 2? I mean, I know we talked about doing a simple metadata on race and ethnicity. Can we also add some kind of, I don't know what people think about this. Can we also add some kind of survey as to what ethnicities are already available and where the holes are in terms of diversity? So some sort of survey of what's available across all the different repositories. Because that might identify, for example, we may need to up our efforts in Brazil, for example, or South America to bring in more diversity for these data sets. And I know it may not be useful now since we do all have a lot of, the data is still sparse across almost every ethnicity, but it would be really interesting to have a kind of a long-term constant survey going on as to how diverse our data sets are on an ongoing basis, like at least once a year come back and saying, you know, just have an effort on a survey or a report on genetic diversity inside of these repositories.
Yeah, what should I write? What do you?
So I would maybe say, maybe as a last thing, number C would be either a task force or a report. It doesn't have to be task force, it sounds too laborious, but something like a report, a yearly report or survey on the diversity efforts, on the data diversity efforts inside of HCA, or not just HCA, all the repositories. Because if we don't know as scientists that we're not getting enough, you know, American Indian.
I mean, I would suggest,
just
looking at the time, should we now spend the last, next 7 minutes or so, think about a bit somatic variation, then go to this blue box, because we were so primed to fill in that blue box, which is probably, what can we concretely take away from this? That we have a good half hour to think about the blue box, that we maybe a bit quick on somatic variation and then move there. Would that be all right?
Yeah.
Yeah. So what was the somatic variation we discussed? Wasn't it the first point, sort of methods and approaches for extracting or for genotyping single cells for an known or well, for detecting, I think we would, let's say for identifying somatic aberrations. And there's probably two approaches. One of them is genotyping known variants, e.g. identified from bulk, matched bulk sequencing. I mean, that's definitely in cancer the way to go, probably, right? I mean, or at least it's a way. And then it's also a de novo discovery of somatic aberrations from single cell data only, yeah? And that's, I guess across all assays, yeah, from SE, RNA attack, DNA, etc. Is that roughly, you know, probably that's what we had in mind, right? Was there anything else with somatic variation? There were some interesting points I don't remember.
Well, there's a physiological, like Chris Walsh had done that study that showed that some diseases may be linked to, I don't know if that's what you were thinking of, but some diseases may be linked to local somatic variation in tissues or organs. I don't know if that's...
You're saying maybe to do this in, so a lot of the HCAs in healthy tissues, but to also propose to do this in disease versus healthy tissues?
Yes, that would be.
Mm-hmm.
In a disease context as well, right? I mean, one could also profile.
We also don't have there's been different opinions about how much somatic variation there is in normal tissue. Like even with mitochondria, because that's an extreme example because it has a lot of reactive oxygen species around the DNA, but this heteroplasmy, a lot of heteroplasmy in tissues in mitochondria. So that's another source of variation is mitochondrial heteroplasmy. But anyways, we don't know in the germline how much variation we have from cell to cell. I mean, I don't think anyone's done a really deep survey on that.
I think it depends greatly on the tissue, right? I mean, if you do skin,
Yes.
a lot, and esophagus, and you know, the brain is probably quite little. So
yeah, but I mean, what is normal, right? So if we had a biopsy from a patient and we say, geez, you've got a lot of somatic variation, that's bad, or is that you have a lot of somatic variation that's normal? So that's what you just said is a great point, is like, do we have somatic variation changing a lot from tissue to tissue?
Yeah, and also so some genetic mosaicism studies, and also for cancer study specifically looking at adjacent tumor adjacent tissues and how clonal expansion was also already happening there. And I think currently, so because of the technology for limitation and for detecting point mutations, CNV might be a better approach in which you can even do from single nucleus DNA sequencing, and also in some way from tumor transcriptome, single cell transcriptome profiles, you can just combine like 100 genes as one block, and then like a little low resolution, but you can actually kind of extract kind of copy number variations.
yeah, I mean, CNVs make a lot of sense in cancer, right? In sort of aging of healthy tissues, that's not that much, I guess, most likely. So, but I think it makes total sense. I mean, that's sort of, we need methods to sort of get that variation out, and I think there are some interesting, you know, directions, and the mitochondria components also I think very interesting. So, what do you want to recommend? Anything else? I mean, to sample more material. I mean, we said sort of assaying normal variation ranges, you know, using single cell assays. I mean, one could do combine single cell DNA, RNA, and I don't know. I could also say one reanalyze existing data. I mean, well, how about that? We could say, you know, survey, reprocess HCA data and other open resources. To search for somatic changes, recurrent maybe. One element would be like mutation signatures, right? I mean, you know, the sort of cancer world and look at mutational processes e.g. linked to aging. I don't know. Just could do that.
Kind of need an encode for single cells.
Say again? We
kind of need like an encode for single cells, you know, looking at all the functional, every last bit of functional, like a project that goes out of its way to make sure that it covers every single functional and component that's measurable at this point.
Yeah.
Or have a set of samples that exist that you can reanalyze. I mean, you might not be able to do the same exact cells each time, but you could probably get different parts of the same tissue.
Yeah.
But it would be really, or the same parts of a cell line, you know, that you freeze so that it doesn't change its genotype. It would be really interesting to try to do a large multimodal study. It doesn't have to be large. I mean, it could be a limited number of cell line samples, but just try to look at genetics and like all these modalities at the same time. I'm sure they're probably talking about this too in the multi-omics.
Yeah, I think so, yeah.
Project. But I mean, from a genetics point of view, I mean, they may not be thinking about anything past cancer genetics, and we're thinking about germline as well. But in cancer genetics, obviously, would be great to add in.
Can go back to, right? I mean, that's a kind of go back to. All right. I mean, just looking at the time, should we go and try to figure out when we run this blue box? I think that's probably
yes, that's
the most.
That's good.
Yeah, that sounds good.
What can we solve by collaborating?
There's the diversity aspect.
Yeah, I think one is kind of getting a tally of the different studies that look across multiple individuals and studies that have genotype data, or recommending people to run genotype arrays that are, you know, pretty cheap on the donors of older samples.
And augmenting existing studies by a genetic diversity component. Yeah, I mean, that's basically, I mean, that's a clear one. I mean, should we say we do a genetic meta-analysis of HCA data and similar, and just say, hey, let's just reprocess the data, see which variants you can call, you know, and look at somatic variants and germline variants. It might be a bit of a long shot, but, you know, on the other hand, if you have lots of cells, you will capture quite a bit of the variation, of the coding variation. I mean, maybe there are more, right? I mean, definitely the genetic meta-analysis,
And you're thinking within a tissue, across all the different cells within a tissue, across multiple different studies, or even across tissues?
It's a good question. I mean, taking all blood would be probably a good way to start, you know, but we can do both, you know, within and across tissues. Combined.
There's going to be a lot of some methodology there, you know, to make sure we're accounting for batch effects.
Yeah.
You know, a lot of differences in the experiments and sequencing depth and so on.
Yeah.
Yeah, but I think that's a good ambitious goal.
Yeah. To redefine regulatory landscapes in health and disease. I guess that's sort of what we want to do, right? So, what else?
I think maybe the kind of the axis of looking at genetic regulation across with different phenotypes, different molecular phenotypes.
Yeah, that's studies like we mentioned with CRISPR or against like the LCLs, or is that what you mean, Violet?
Yeah, I was thinking more of studies where they've done, gene expression, RNA-seq, proteomics, metabolomics, but also what you're saying. Yeah, I think that's also. So would it either be within, these multiple assays done on the same samples, which would be ideal, but also done on different samples, but within the same tissue? We could start thinking and developing methods for comparing genetic regulation with different molecular cellular phenotypes.
Okay, so beyond RNA, yeah? I mean, and ideally, something you can go back to, because then you can just add stuff, maybe reference, maybe how we call this, establishment of reference. Resources. Attack, proteomics, spatial. All right, I think that's good. Anything else?
Did that sentence go away? It looks like it went away. Is there still a number 3? My number 3, okay. Because the final template, it disappeared. Oh, it went down to the next page. Okay. So the other thing, can we add that maybe a section on what we mentioned about doing the working together to try to characterize genetic perturbations? Like when we were talking about CRISPR, et cetera, because there could be people, I don't know what people think about that, but there are resources for that with, people have repositories for different genetic problems or doing CRISPR studies, because that would kind of allow us to look at the genetic effects on cell type. I don't know what you guys think about adding that.
You want to get at this idea of, using genetic perturbations for cell type identity definition, or?
Yeah, for cell identity and cell state.
Mm-hmm.
So what the effect is of a genetic perturbation, either native to the person that we're getting the samples from or by CRISPR, with the genetic, these would be specific disease genes. It wouldn't just be some random location, but how those would affect cell state and cell type, basically normal function.
Should it be done in the context of animal model or organoids to be able to see systematic genetic changes and then see interaction with different cell types?
Exactly, yeah. See organoids or animal models.
Yeah, for 4, I think that's very interesting. And then just to compare to the cell identities from, classical RNA-seq or other, like ATAC-seq, other phenotypes.
Yeah, versus observational data. Should we just say that as assays?
Well, it's not really just using genetics as a tool to identify cell identity as much as it's using genetic perturbation to understand the effects of these genetic changes on cell identity. I guess
I think that's another point.
Yeah.
It's a
both
different.
of them.
Yeah.
I mean, they may be different. Maybe you don't want to. Yeah. Because they're both one.
You're muted.
Sorry.
We don't hear you.
Yeah. One is a population level question, right? Which is what we've been talking about. And one is more of a specific, I mean, using rare diseases that are otherwise, I mean, they're not so rare that you don't get multiple people with them, but using different diseases to understand how genetics perturbs cell state and cell type.
Yeah. Okay, recommendations. What should we want to do? I mean, I guess we could now really go back to these individual points, right? I mean, 1 to 5, or as many as we can make specific. So,
one of them would be recommending that the different atlases include race and ethnicity.
okay.
Because that's an action item they can do right away. I mean, if they have it, if they're collecting that information.
So we would say we recommend to incorporate metadata and genetic data into ingest processes, right? I mean, that's
or even including race, and sometimes just metadata on race and ethnicity. Even if we, or metadata on race and ethnicity. I'd be a little bit specific there because
yeah, you're right. Yeah.
They won't understand because they weren't in the conversation. Yeah. Very good.
I mean, are there existing cohorts one could tap into, or that one could bring together, or one could, join things? I mean, another dimension is, would it make sense to say, oh, there are different cohorts that now do some single cell genetic analysis, and one can bring them together to create such resources? I don't know.
So that idea of trying to do a survey, I'm not saying that that's the perfect idea, but just understanding what resources everyone has is going to be important. Is there any way, maybe that's an action item we can also do soon, as to do I hate to use the word survey. What's the word that would be like a, there you go, that's a good one.
Starting by surveying existing efforts. I think that's
good.
Okay.
We talked about having a second level of, the Human Cell Atlas itself has already planned for a second level of data protection to include genetic studies that would, or protected data, not just genetic studies, protected data that would enable more genetic studies to be done. So I'm not sure if there's a protocol or I don't know this question, so I don't know if anyone else knows this answer, but if there's any kind of protocol in trying to bring in more studies into the HCA that would be able to enable us to do these genetic studies better. So like, how do we encourage people to start thinking about doing this with their IRB and hosting it locally, hosting it so that the HCA can, who's going to manage the data? So like if I go to my IRB and say, I want to do a genetic study with single cell data, and then I want to give it to the Human Cell Atlas, they're not going to like that. So I would love to know what the protocol would be to try to get data from other repositories into some sort of managed. I don't think the Human Cell Atlas may directly have to manage the data. It could be managed at a distant repository, but maybe it should be listed at the Human Cell Atlas as an existing genetic data set associated with the single cell data set. So there's a bunch of different ways to do this, but I'm not sure if I can share data with an external repository unless we came in with a protocol that suggested that in the first place. So in order for all this to happen, there has to be some sort of way to control the genetic data. And I'm just not aware of how that's going to be done. So maybe that's just my ignorance talking.
Yeah, I mean, I guess, you said an important element, right? That Controlled access will probably also include just disease status. You know, as soon as you have disease status, you can also get genetics in, I guess. That's probably, from an IRB's perspective, is somewhat similar.
Right.
You know, I mean, it depends also on the IRBs. Yeah, but I guess it's a support structure, you know, facilitate that. Yeah, I mean, there might be also an interface, you know, with existing data ending up in dbGaP. There's also a question to avoid fragmentation. I don't know whether one wants to.
It's already fragmented at Kids First. Even though dbGaP manages the metadata, we manage all the data in a separate repository. dbGaP's not really taking in any more large studies, which is unfortunate.
Oh, really?
Well, I mean, they're taking in like summary data, but in Kids First, we have to manage all the large scale data. We do it through a shared key, like Auth0 with dbGaP. So dbGaP will give out the key and they'll manage the data use committees, but we have to go and manage the data export to the data release and the data storage. So I'm not saying, so that's another way that HCA might be able to work with dbGaP, right? Is something like Terra or one of these other platforms that they use, Anvil. One of those could manage the data for dbGaP. Because the Kids First is a common fund project. And there's no reason that we're not special, right? So there's no reason that any other large scale NIH repository couldn't broker agreement with like the Human Cell Atlas to manage some controlled genetic data for release. But that would have to be built into the protocol to start. So maybe that's a recommendation we can do is there a way that we can integrate with dbGaP through not just the HCA, but all the projects. And I'm sure HubMap is already there. But are there other external projects besides HCA and HubMap that could also partner with dbGaP to release these data? I don't know.
Yep. All right. I think that's a good point. I just merged into it.
It's a mess.
Okay. 40 minutes ago. Let's get to number 2, maybe. What do you think? Genetic meta-analysis within and tissues to find relevance with healthy tissues. I mean, that would be basically, you know, that the DCP, where the data coordination platform, that they basically read genotype, you know, call genotypes and all that they include this in the pipelines. Is that a recommendation?
It would be really nice for them to do that, because it wouldn't be all that difficult for them to call genotypes from even just from the RNA-seq data and provide that as an additional file so that people don't have to go and redo all the analyses. If there's a standard for that, a standardized pipeline for that, you know, could be an additional file that gives you whatever they were able to detect. Obviously it wouldn't be perfect, but wouldn't hurt. And that's something that could be done right away without additional data needing to be generated. That's a fantastic idea.
I guess one could also have best practices, right? I mean, you know, and then do those, you know, incorporate best practice workflows, you know, to deliver added value to existing studies. All right. And then genotyping, that's both germline and somatic, right? I guess you can just do both in one go.
Yep.
All right. Anything else in this section?
We were talking about, no, I think that's right.
There's also metadata, you know, yeah, this is also recommendation to metadata working group. You can maybe too, right? I mean, essentially the metadata working group has to do that. To incorporate. Okay. Anything else for 2, or should we just move on?
Is there something needed for, because you talked about within and across tissues, so should there be some recommendation to kind of organizing that effort?
It's a good question. I mean, how about analysis or consortia cross HCA working group to define or derive regulatory maps initially within large tissues, e.g. blood, later across tissues, to be more specific? Is that what you, something like that? Yeah, you're right. I mean, otherwise we probably or initiate pan HCA, whatever, or HCA, NIH, whatever. Yeah, I mean, obviously, I'm just adding all the rest to it as well. 3. Shall we go to 3 just to let me get through? We have 10 minutes left, and
well, we have the opportunity. Well, I think also what you, building off what you just said, real quick. With their when they do this analysis, we might be able to detect somatic variation just within the RNA-seq data per tissue. And number 3. So, I mean, and 2, it really would be 2B, but I mean, remember we were talking about, I think that's what she was just saying, is that we had multiple variations in somatic variation between cells, or cell level somatic variation, or tissue level somatic variation.
I mean, that's basically what you mentioned, right? That we basically lesion profiles and characteristics,
cell types and tissues,
and tissues.
Yeah, sorry, yep.
I agree with that. Yeah, particularly to do cell types, the same cell type with different tissues. I think there's a lot of stuff in there. Yeah, I think that's great.
Yep.
And then for in terms of methods, the approach should be different in cases where you have genotype data from the blood, and in the case you don't.
Yeah, the best practices would be, definitely different.
Okay, yeah, with and without genotype reference.
Yeah.
Like that? Does that cover it? Yeah, so 3, I don't know what that means. I mean, that maybe we have to look at some existing resources. I don't know who has the best resources for that, right? I mean, I guess what you were mentioning is that you want a big cell bank where for lots of people, and you can order material and, you know, throw your favorite assay at it. Yeah? And the easiest one would be some sort of cancer cell line reference database, LCLs or IPS-derived things.
It certainly would help methods development.
Yes.
It really would for the people who are doing this kind of thing to have some sort of reference.
Reference cell bank for single cell
genotypes, and to get like a genetic reference bank, kind of like they do for the genome in a bottle.
Yeah, I agree with that. Yeah. So it's define most suitable system. I guess that could be e.g. Cell line, IPS derived. Anything else that really makes sense? Probably that's it, right? I mean, it needs to be either IPS derived or cell line, or IPS derived organoid. I guess organoid would be great, because it has a spatial.
Okay. Can you mention that it would be something like a standard reference? The word like standard reference or repository for that standard reference. I mean, NIH could do that, for example. Or FDA or somebody.
Okay, I mean, here we do working group
Yeah.
Working group to define most suitable system. How do you call this? To define standard reference?
Standard reference, or most suitable system, or standard reference for a standard reference, establishing a standard reference or set of references. It could be more than one, but just allowing people to always access the same predictable genotype / cell line. Because usually with these standard references, it requires repeated periodic genotyping to make sure that the genotype of the cell line isn't drifting, and that you have a standard reference that you can go and test these methods against. Because we're coming up with so many methods now that are including all these multimodalities that it would be really nice to have that. And they may be talking about this in the other meeting, but.
No, I agree with that. Yeah, it's a working group, and and then you need a distribution center and all that. Set up, open or create first open access community data set with genotype RNA attack ?
Mm-hmm.
That people jump on it to foster methods development in that space. Community uptake,
and keep a distribution center for the system for keeping those samples and keeping the sample integrity and shipping it out to people who want to retest as new modalities are invented for genetic studies.
Yeah.
Sounds great.
Similar to HapMap.
Yes.
Not the all times.
Exactly. No, that's right. Exactly. HapMap is a great example.
Yeah. Okay, I think this is a really, really good point. I'm running out of juice, but we are almost done. How about 4 and 5?
So you want me to start typing number 5, or I mean, people can add to it or something, or because I mean, number 5 would be simply, create a working group or a group that's interested in looking at the effects of genetic perturbations. It really goes across tissues. Maybe people want to focus on tissues, but this kind of question in number 5, at least it seems to be pan-tissue approaches. So this would be really a protocol development is number 5 for me. Would be like protocol development for animal models, CRISPR organoids, because there aren't standard. Maybe we could have a protocol group or something to suggest how to do that, or maybe it's going to be too tissue specific. I don't know. And yeah.
I think it's a really cool thing. I just don't know what's the consortium level working together approach to tackling it is.
Right. Yeah, I'm not sure.
The other ones I can see, that like, a big NIH-HCA, there's sort of something like it's actionable for 5. I don't know what to write. Same actually for 4. I don't know.
Maybe it's a space where people that are interested in have a system that they're planning on modeling some rare disease, where they can interact and share what are the protocols they're using, both experimentally and the analyses. People can start kind of comparing notes and maybe start to build some recommendations or common practices. Obviously, from different tissues and different diseases, it might be a bit different. But I don't know, to have some space where people can start kind of sharing notes and discussing what can be. For example, at the Ocular Genomic Institute where I work, there's some PIs that are starting to set up experiments in retina organoids to look at the effect of mutations that lead to rare inherited retina degeneration diseases, and do single cell RNA-seq on organoids. And I'm sure there's many centers around the world, doing similar things for different rare diseases. So maybe having some forum, or initially, I guess, people that have already some ongoing project that could get together and share kind of protocols, experience.
I mean, I think there are. I think I like the community portal and protocol exchange. You know, there's even something that could be quite fun is normal variation panels for canonical systems that are used in rare disease genetics. You know, I mean, if you have your favorite whatever mutation you want to assay, yeah, in developmental, I don't know, neuronal cells from iPS differentiation, you'll have a lot more power if there's a big reference cohort where someone has done hundreds of people, normal reference people, and you can compare to that. But that's only possible if the protocol is matching, right? There's a lot more powerful when everybody does their 5 sample normal versus the 1 disease sample. So that could actually be quite an interesting component. I mean, just listening to your suggestion, maybe to add that as well, you know, normal variation panels for canonical systems, you know.
I agree. I think that's a good suggestion.
EP IPS derived, you know, to reduce entry barrier and increase power. I mean, that's I guess, I think that could be really useful, you know. So we started that, and you have a reference panel of sort of neuronal differentiation of 200 people, and other people, you know, with rare disease, they just basically, you know, differentiated one line, and you know, that was a lot less work, and you know, to really look at aberrant expression patterns of a particular disease. So I think that's actually maybe this is
that's a really good idea.
Okay, 4. Do we want to write something, or genetics as a tool? I don't know. I mean, I think it would be cool to have a system where you have different cell types, and we throw all sorts of assays and approaches that it refine cell identity. But I'm not sure this is really for this sort of working group to drive, right? It should be more, you know, it needs to be multi-omics. And
I think there is a cell identity clustering working group. I think later this afternoon. Yeah, I think it's part. I think genetic regulation. We should at least raise the awareness that genetic regulation of gene expression and other modalities should be considered as well as one of the mechanisms for defining cell identity and a cell state, cell type in the comparisons with other modalities. Maybe we can just make a note here that this might be part of a cell identity working group, but that this should be considered.
Yeah, it's just more a reach out thing. I agree with that. Yeah. Okay.
Great idea.
All right. We are 2 minutes past. Shall we wrap up? I mean, I think we need to.
Yeah, we have to break for lunch. Yep. Let people answer their emails.
Yeah, but this has been fun. I think, you know, we have been a small group, but I think somewhere it's almost easier to move if you know, have a small discussion. I really enjoyed that. I mean, I thought we got to something. I didn't know where this would lead to, but I like these things. It would be fun to do something.
Yeah, this is fun stuff.
Yeah, it was a great discussion.
And so remember, we can add things to the blue. They encouraged us last time to add more stuff to the blue boxes before 8 p.m. tonight, right? So if there's anything else, anybody else comes up with.
Yeah. One email contact. Well, I mean, we can write all our emails there. We're not that many.
Yeah. I'll type in mine.
Well, I don't know what I would write from this. Maybe we can write here about what we do. I don't know. What else do we need? Money. Oh,
I would say, yeah, money and more communication. I mean, communication support, like community support would be nice, because that's where it would start, right?
Is there a Slack channel or something within HCA that that's open for collaboration?
We can make one.
Yeah.
That'd be a great. That's a great idea. I have it open right here. What do you want to call it? We could call it. You want to just call it genetics? It's pretty broad. Does anyone have any votes?
Single cell genetics.
Yeah, that works.
Right. Should I call it single cell genetics or just okay, single cell genetics?
It's catchy.
Okay, so if you just look for channels, it's called single cell genetics. Here. It's, where are we? Yeah, it's just that. I put it in chat, but that's what it's called in the HCA. And I didn't make it private, so you should be able to join it.
Good.
Just mention that we created the Slack channel. Maybe that's not in there. I haven't even looked at the.
How do I join this?
I think you just go up to, if you're in Slack in the Human Cell Atlas, you go to channels and you click on the channel. Just don't click on the plus, but click on the channels itself. The name channels, right? And you should have a browse channels there.
Okay.
And type in single cell genetics.
I got it.
And you should be able to join.
Thank you.
So this is not a new workspace, but a channel?
It's a channel in the Human Cell Atlas. So you belong to the Human Cell Atlas Slack? There's a Human Cell Atlas Slack channel, the whole thing.
Gotcha.
Not channel, but a space. There we go.
Okay.
Slack space. You should be able to join it.
Yep, thanks a lot. So this is called single cell genetics on
single cell genetics. Where's the yeah.
All right, cool. That was fun. Well, very nice meeting you.